Hello, Year 10 triple scientists. Welcome to the Plant 2 topic. In this topic, it follows straight on from the Plants 1 topic that you may have done in Year 9. So that may feel like quite a long time ago since you learned that last bit of information. In this topic, we are looking at plant disease, plant growth, plant hormones, and deficiencies in the plant, as well as those that require practical as well. Without further ado, lesson 1, we are looking at the question, do plants get ill? At the top of your page, please can you write the driving question, which is, do plants get ill? The reason that I'd like you to do this is just to keep your notes nice and concise. When you look back, you'll be able to see exactly where you've done the work for lesson 1. Just makes sense. Keep yourself a bit more organised. The keywords for today's lesson are pathogen, communicable, biodiversity, disease, virus, bacteria, fungus, protozoa, famine, yield, and monoclonal antibodies. Now, with the triple science topics, a lot of the time there is overlap with other topics. So you may notice monoclonal antibodies crops up in another topic. Communicable disease, meaning a disease that can be caught or passed on, as well as pathogen, which is the microorganism that causes disease, as well as virus, bacteria, fungus, and protozoa being examples of pathogens. That's obviously from the infection and response topic. Okay, so again, there's lots of overlap. We will tackle these keywords when we come to them. So why are we learning about plants? Well, almost every ecosystem and animal, including humans, depends on plants for energy. By understanding how plants work, we can improve the yield of crops and help protect our environment. Right, so some starter questions for you now. You don't need to write down your answers. If you do, just do it on some scrap paper, perhaps down the side, just to get you thinking. You don't have to write down your answers. What is a communicable disease? What are pathogens? What are the 4 types of pathogen? And can you give an example of each type of pathogen or the disease that they cause? So this is a bit of recap really from the work before. Please pause the video, and when you come back, I'll go through some answers. Hello, welcome back. Communicable disease is a disease that can be passed on by pathogens. So what are pathogens? They are living organisms that infect hosts and cause disease. Now a host would be a living thing that has the pathogen inside it that causes the disease inside them. The 4 types of pathogen that we looked at in the infection and response unit are bacteria, fungi, funguses, virus, protozoa. Some examples of those: salmonella and gonorrhea are examples of diseases caused by bacteria. Athlete's foot is an example caused by a fungus. Measles and HIV are caused by a virus, as well as and malaria is caused by a protozoa. So there we are, a bit of recap. So why do we have to learn about plant diseases then? Well, plant pathogens kill or reduce the growth of many plants, which can affect biodiversity. Biodiversity being just the amount of different species that there are, and with increased biodiversity, then you can have much more healthy ecosystems. So like think about the rainforest compared to a field of just a single crop of just one species. Plant pathogens of food crops are especially serious when they affect the yield of crops that people rely on. So a really excellent example is the Irish potato famine, which I'm sure everybody will have heard about. This happened in 1850. This caused the death of a large number of people through starvation. Literally, about 1 million Irish people starved to death because the potatoes could not be eaten. The pathogen caused a blight. This is almost always talked about with fungal infections of plants. But this is an infection that discolors the leaves, but it caused the tubers, which are potatoes, to rot. And so 1 million people starved to death. The plant diseases that we need to know about in detail for the AQA syllabus are tobacco mosaic virus, or TMV, rose black spot, and aphids. So TMV, I suppose, in a similar way to HIV, being a virus, has been shortened down to just the letters. Tobacco mosaic virus is what TMV stands for. Rose black spot is this picture down here. It is black spots that appear on roses, again, not too hard to remember. And aphids, which some of you will already recognise as these little green insects that although are not pathogens because they are not microorganisms, are certainly parasitic. Okay, so in detail now, let's have a look at tobacco mosaic virus. The first word in tobacco mosaic virus is the word tobacco. Now, lots of you will have guessed that this actually infects the tobacco plant. And so some of you might be thinking, so why do we have to learn about tobacco? Like, only people who smoke will worry about this. Well, actually, it doesn't just infect the tobacco plants, but also other closely related species like tomatoes and peppers can be infected by the same virus. It is transmitted, that means it can be passed on, by physical contact between plants, but also when animals or farmers perhaps move between the plants, maybe touching the leaves of some and then touching the leaves of others, the virus can be transmitted that way. Why is it called tobacco mosaic virus? Well, it infects the chloroplasts of plant leaves. This changes the colour of them. As we know, it's the chlorophyll inside the chloroplast that make plants green. If it affects the chloroplasts, then their colour changes. It goes from green to kind of a yellow or white in a mosaic pattern. So again, a mosaic is usually a piece of art made up of like a collage of tile pieces, really. And actually, it will have this pattern, and in a similar way, the leaves become patterned like a mosaic. Another symptom could be that it makes the leaves crinkled or curled up. We'll talk about why this is bad in a second, but there is no cure. So farmers' only option is either to prevent them being infected in the first place, or identify and reduce the spread as quick as they can. Often it will just mean that if there is an infected plant, you just burn it. You just burn the plant. Happens all the time in orchards. If there's a virus affecting one of your apple trees, you burn the tree down. There's nothing you can do. Pretty scary, really. Why are crinkly and mosaic patterned pale leaves bad? Well, hopefully we can link this back to the chemical reaction photosynthesis that we learnt about in the plants 1 topic. This reduces the plant's ability to photosynthesize, which means it cannot grow. This will reduce the farmer's yield. Hopefully you will remember that photosynthesis, which is an endothermic reaction to make sugar. The point is making sugar, which the plant can use for growth, repair, and actually respiration. It requires water and carbon dioxide. Carbon dioxide gets from the air, water from the soil, and it makes glucose, which is this carbohydrate, and oxygen as a waste product as well. So if there's not enough chloroplasts or chlorophyll because of this virus, then the plant cannot do photosynthesis, so it can't make glucose, so it can't respire, and it can't store any energy, which is kind of what you want your crops to be doing, so you can pinch them and cook them up. Okay, so the second plant disease, is rose black spot. Again, there's clues in the name to what it does and how to remember it. It looks a bit like this. It is caused this time by a fungus, not by a virus, and this infects roses, again in the name. What it does is it infects the leaves again, and it causes black or purple spots to appear on the leaves. This is in fact necrosis, which actually just means death, plant cell death. So the rest of the leaves often turn yellow and can drop off. So again, if the leaves aren't working, this reduces the plant's ability to photosynthesize, so the plant will not grow and you can't get a crop out of it. How is it transmitted? Well, actually, this time it can be transmitted over the air or in water, as well as direct contact from gardeners. So fungi produce spores, which can be microscopic, um, effectively offspring, basically, and they spread those just in the air, millions and millions and billions of them, and they will just grow wherever they land. It can be transmitted in air or water as well as direct contact through gardeners, and is treatable using fungicides, which is like a pesticide but for funguses, and by removing and destroying the infected leaves so it cannot spore anymore. That's black rose spot. Again, black spots upon your rose. Right, the last one is caused by insects, specifically aphids. So an aphid, bit of a weird word, aphids, are these tiny but not microscopic insects. Okay, they're usually green, they've got some wings. Aphids are common examples of insects that infect plants, seen in the UK gardens and greenhouses. In particular, they infest roses and tomatoes. They're found in large numbers on new shoots from which they suck sap. So they have like a hypodermic needle in their mouths, which they use to stab into the plant, specifically into the phloem inside the stems, and they suck the glucose, the sucrose, they suck the sugars that the plant is using. Now, one aphid on a plant might not have much impact, but actually, when there's one, there may be loads. And actually, this will reduce the growth rate of a plant and can actually kill the plant by just sucking them dry, literally. Aphids are eaten by ladybirds. And so, a gardener often will try to cultivate ladybirds in their gardens, and it can be used as a natural way to reduce aphids. So, the more ladybirds you have in your garden, the less aphids you have, and therefore, hopefully, the less plant death you're going to have. So, this is called a biological pesticide, by using a biological creature like a ladybird to kill and eat the aphids. So, those are the three there. There are a small selection of other diseases that you need to be able to identify. Now, you don't need to know them in the same detail as tobacco mosaic virus, black rose black spot, or the aphids, but you do just need to know, you do need to be able to recognize these. So, given the symptoms, you might be able to say what they are. So, mealybugs, which again is a bug that lives and damages a plant, and it causes stunted growth. Again, instead of black spots on a rose, you might see decay in other places caused by a fungus. This is called the rice blast fungus. You may have malformed stems or leaves caused by the ash dieback fungus. Okay, so just three more that you may have to identify, but you do not need to know in detail. Okay, quick to your knowledge. I need you to match the disease to the symptoms. Basically, down the left-hand side, you've got the three diseases that we talked about. Down the side are the other side of the symptoms. Can you match them together? Pause the video and just connect the numbers to the letters. Okay, welcome back, year 10. Let's have a look. So the answers are tobacco mosaic causes crinkled leaves, mosaic pattern, and missing green parts of the leaves. Rose black spot causes dark, dead patches on the leaves, and aphids reduce the growth rate. And it's very obvious because you can literally see insects on the other sides of the leaves. Okay, again, quick bit of a quiz. Can you finish the sentence? Tobacco mosaic virus is a problem for farmers because. Is it A, B, C, or D? Have a read of those boxes, pause the video, and we'll go through the answers when you come back. Okay, the answer is A. It affects chloroplast, so the plant doesn't photosynthesize and grow. If it doesn't photosynthesize and grow, you won't get a higher yield, and the farmer will get less money. Right. So, how can we identify plant diseases? How do we know that they have them? Now, there are a number of ways that, again, you do need to know, and this is in the list of things in AQA that you do need to know. What can you do about it? Well, you can use a book or even the internet to identify plant diseases. You can take a small cutting of an infected plant, give it to a garden centre perhaps, and the staff will often be able to identify and treat the disease. So take it to an expert. And the last one is actually using monoclonal antibodies. This can be quite complex, and you go into a lot of detail in another unit, just for triple scientists, but we will go through a little bit of detail now. So those that are more difficult to identify, you may take cuttings of the plant, which can be analysed by scientists in laboratories using testing kits containing monoclonal antibodies. So very quickly, how do monoclonal antibodies work? Well, a monoclonal antibody can recognise a specific antigen. Samples of the plant tissue can be tested with the monoclonal antibody. If the pathogen's antigens are present on the plant tissue, then the monoclonal antibody will stick to it. Another antibody is then added which can bind to the monoclonal antibody. This antibody has a marker on it, such as a dye, which then signals to the scientists that the monoclonal antibody has recognised the antigen, and therefore the disease is present. So, how does that work? A monoclonal antibody is basically a special type of antibody that has been cloned and will stick specifically only to one antigen. Antigen are really proteins that appear on the outside of cells. Now this says a cancer cell, but this could be a disease cell in a plant, and it's got all of these antigens on the outside. Now the antigen on the outside may be present if the cell has a disease, so the antigens may change. Scientists produce antibodies that stick specifically to the antigens caused by that appear when you have the disease. And so what happens is the specific antibodies, excuse my animation, stick to the antigens on the cell, and once they've stuck, they can actually have these radioactive markers on them, which can then signal and say, actually, yes, we have stuck. So this has applications even in pregnancy tests. This is how those work. We will go into more detail into monoclonal antibodies another time, but that's just a quick run through how it works. You have antibodies that stick to antigens and identify whether or not the disease is present. Okay, now I want to hand it over to you. This is the point of the lesson normally where I'd ask you to discuss the answer of a question with your neighbour. By discussing something, you're actually kind of rearranging the thoughts in your brain and actually can stick into your long-term memory a lot easier. Instead of actually explaining to your neighbour, however, because we are not in the lab, I would like you to literally get up out of your chair and go and tell somebody the answer to this question. Could be parent, it could be brother, sister, it could be your pet, it could be your neighbour over the fence. Doesn't matter, as long as you're telling somebody the answer. And I would like you to tell them, what are the ways you could identify a plant disease? Just been through those. Get up and go tell somebody. We'll go through the answers when you come back. Pause the video and go tell somebody the answer. Okay, welcome back. Year 10. What are the ways you can identify plant disease? Well, look them up in books or use the internet. Take a small cutting of an infected plant or a photograph and take it to a local garden centre. They may have staff that can help. And you could use monoclonal antibodies, where scientists can identify the diseases using monoclonal antibodies. Right, now it is time for the writing phase. So underneath the driving question, which you should have at the top, which is, do plants get ill? I would like you to work your way through these objectives as questions. The keywords are at the bottom, which you are going to need to use in your answers. The first question, could you label it the describe section? And the first question is, can you name 3 examples of plant disease? Can you remember those? The second question, the explain section, is can you explain how mosaic virus, black spot, or rose black spot, and aphids cause plant disease? The use section, can you please answer the question, explain a range of methods for detecting plant disease? And finally, your combined section, can you explain how monoclonal antibodies could be used to detect plant disease? So 4 different questions. Can you title them explain, describe, use, and combine? And give yourself, between 10 and 15 minutes for this. When we come back, we'll go through the answers of those. Good luck, year 10. See you soon. Okay, welcome back. Well done on that, by the way. 10 minutes of working can be quite draining, can't it? Right, time to go through the answers. Now, this would be the point in the lesson where we would get a green pen and go through and mark. If you do not have a green pen handy, just use a pen or pencil of a different colour. That should just be fine. Now, it's important not just to give yourself ticks for when you get things correct, and it is important to do that. I'd like you to add in the extra detail that you're missing. So really importantly, if you got something wrong or maybe you've missed something, now is your opportunity to fix that on your page. Okay, so the 3 diseases that we looked at before are tobacco mosaic virus, or TMV, rose black spot fungus, and aphids. More could be mealybugs, rice blast fungus, or ash dieback. These were the 3 that we looked at in detail, and those were the ones I was looking for, really. The explain section was can you explain how they cause disease? Well, tobacco mosaic virus affects chloroplasts, which turns the leaves yellow or white in this mosaic pattern. It causes stunted growth because the plant can't photosynthesize. It's transferred or is caught through physical contact, and there is no treatment. The rose black spot fungus causes leaves to turn yellow or black and drop off. It causes stunted growth, again, through lack of photosynthesis, can be transmitted through air or water or contact, and can be treated by fungicides. And aphids, why do they cause disease? Well, they suck sap from the new shoots. They reduce growth rate and eventually will kill plants, and can be treated by predatory insects like ladybirds. Right. The yellow section, the use section, was how can you identify the diseases? Well, books or the internet, taking cuttings and showing them to garden centres or experts, and cuttings can be analysed by scientists where they're tested on using monoclonal antibodies. So hopefully you've got those. And then finally, quick explanation of how monoclonal antibodies work. Again, this will be easier when you have completed the unit where we do look at monoclonal antibodies. Monoclonal antibodies can recognize a specific antigen. Samples of the plant tissue can be tested with the monoclonal antibody. If the pathogen's antigens are present in the plant tissue, then the monoclonal antibody sticks to it. Another antibody is then added which will bind to the first monoclonal antibody. This antibody has a marker on it, such as a dye, which will then signal to the scientists that the monoclonal antibody recognizes the antigen. You recognize the antigen, you recognize the pathogen. If you can see that the pathogen's there now, you can say what disease there is. Okay, as always, time to write your today I achieved sentence. Can you please write, today I have achieved a describe, explain, use, or combine because I can. It's important that you actually write because I can and say why. What I'd like you to write after this is either name examples of plant disease, explain how mosaic virus, black spot, and aphids cause plant disease, explain a range of methods for detecting plant disease, or explain how monoclonal antibodies could be used to detect plant disease. We've had lots of practice doing this so far. I don't think we can have much trouble with it now, but make sure that you have got your today I've achieved sentence at the end. Thank you. Okay. As always, there is a 10-question Show My Homework quiz that will be on Show My Homework now. That will both help you recap some of the information that you've learnt today, but also show us how well you're doing. Okay, thank you very much, Year 10. I'll see you next time. I think we're looking at plant deficiencies and how that affects the plants. Okay, bye for now.